Biotechnology. Polyethylene glycol transfer gene without a vector. Cohen and Boyer, 1972. First rDNA Salmonella typhimurium. Helicase break hydrogen bond during DNA replication. rDNA Cohen Boyer. Vector clone only small fragment of DNA. Plasmid. Vector plasmid that transfer foreign DNA into the living cell. tDNA Agrobacterium tumefaciens. Plasmid used as a vector in genetic engineering. Ability to carry foreign genes. Plasmid are suitable vector. Small circular DNA plus their own origin of replication site. Technique of deliberate manipulation of genes transfer to different organism. Genetic engineering. GARD TTC E. coli. Bacteria protect themselves from viruses using endonuclease restriction enzyme. RNA polymerase, RNA primer. Roman number indicates order of discovered from sources. DNA fragment separated by electrophoresis. Restriction enzyme used for cutting any DNA fragments. Staggering cuts leaving streaky ends. E. coli, Hind3, BAM, HI. DNA molecules to which a gene of interest is integrated for clone vector. Dicotyledonous plants from crown gall, specific part of DNA from TI plasmid. GARD TTC CTGC gets integrated with plant's chromosomes. Ejection site. Antibiotic used for select transformants and recombinant bacteria. Recombinant DNA bearing penicillium restriction site. Transfer growth and no transfer die. Non-transformants die. Transformants will grow and non-transformants will die. Ampicillin tetracycline antibiotic resistance gene. Used as a vector for transfer DNA fragments. Plasmid bacteriophage. Ballistic technique used gene transfer process in plants. Polymerase chain reaction useful in DNA amplification. Trichoderma production of chitinase. Transpons mobile genetic element. Electroporation making transient pores in cell membrane to introduce gene constructs. Source Taq polymerase thermophilic bacterium. Taq bacteria are DNA polymerase. Restriction enzyme are used in isolation of DNA from other macromolecules. Agrobacterium tumefaciens enter host through wound and injuries. cDNA prepared from mRNA. Genomic DNA library is packing of donor DNA in a collection of vectors. Clot formation prevented by TPA. Insulin other than pig not cause immunological problem. Insertion and activation of beta galactosidase. White colonies. Recombinant plasmids. Gene can be inserted by modified viruses. Expression of transgenes. Reporter. RTD restriction endonuclease used widely. Bacterial cell. DNA ligases used to join DNA fragments.